Hello biology students, and welcome to video 6 of the cell unit, where we're going to be talking all about osmosis. Now, osmosis is still an example of passive transport, just like diffusion. So this means, just like diffusion, we're not going to be burning any ATP, so we're not going to use any excess cellular energy, and we're still going to have molecules moving from a high concentration to a low concentration. So if we look at the definition of osmosis, it's the diffusion of water across a membrane down its concentration gradient. So this is almost identical to diffusion. It's the same exact definition, but now it's the diffusion of water. So we're basically going to get water to move from a high concentration of water to a low concentration of water. So to further explain osmosis, let's look at this picture right over here. So what I have a before and after shot of a U-tube. So on the left, I have a U-tube where on the left of the U-tube, there's only a couple of solute molecules. So let's pretend they're sugar. And on the right, I have a very high concentration of sugar in a solution. Now, if we were talking about diffusion, and by the way, I also have a little membrane right in the middle of the two halves. If I was talking about diffusion, in theory, these sugar molecules would move from right to left, from high to low concentration. But it turns out sugar is a really large molecule, and large molecules cannot diffuse across a membrane. And because water is a much smaller molecule, it can cross the membrane through special channels that allow it to cross. So instead of the sugar moving and diffusing, we get instead the water to diffuse. So now, let's look at this zoomed-in picture to understand why the water moves the way it does. So if I look on the left, where I only have a couple of sugar molecules, so that's shown here, I get some of those water molecules to form spheres of hydration around that solute, and I have lots of water molecules that are just free and left over. On the right, where I have a lot of my solute particles, my water molecules have formed lots of spheres of hydration, and I only have a couple of free water molecules. So if we think that all these molecules are moving around and colliding, these ones on the left are much more likely to collide and therefore move across the screen. So the water ends up moving from a high concentration of water, where I have lots of free water molecules, to a low concentration of water, where I only have a couple of free water molecules. So here, water is diffusing from a high concentration of water to a low concentration of water. Now, what's confusing about this, and what students always get tripped up on, is where I have a high concentration of water, I only have a low concentration of solute. And where I have a low concentration of water, I have a high concentration of solute. So it looks kind of backwards, because really the water ends up moving from a low solute area to a high solute area. So if you look here, it's moving low to high when we're talking about solute. But really, it's moving from high to low concentration, and we're talking about the concentration of water. So by definition, this is diffusion, because we're moving from high concentration of water to low concentration of water, even though it's low concentration of solute to high concentration of solute. So, if we have water moving in and out of cells because of osmosis, it's kind of logical that this is going to have a large effect on the cell. So in order to understand how the movement of water affects the cells, we have to understand 3 vocab words. They're all adjectives. The first one's hypertonic. So if I say a solution is hypertonic compared to a cell, that means there is more solute in the solution than there is as in the cell. If I say a cell is hypertonic to a solution, that means there's more solute in the cell. Okay, so you have to specify which one is hypertonic. And that means you have a higher concentration of solute in that thing. If I say a solution and a cell are isotonic, that means they have equal concentrations of solute. And if I say something is hypotonic, I mean it has a lower concentration of solute than whenever I'm comparing it to. So a hypotonic solution would have less solute in the cells that are in it, and a hypotonic cell would have less solute in the solution it's in. So depending on what kind of solution I put cells into, different things are going to happen. So let's look at isotonic first. So here's my little beaker, and let's say I have 4 solute molecules. And here's my little cell, and I also have 4 solute molecules in here. So they have, we'll just pretend they have equal concentrations. So that means we have equal amount of free water molecules on each side, and we're going to get as much water moving in as we do moving out. So the shape of the cell does not change. If I have a hypertonic solution, that means the solution is going to have a higher concentration of solute than the cell itself. So let's say it has tons and tons and tons and tons of solute. And then I have my little cell here that has almost no solute in it. That means there's more free water inside the cell. There's less solute, there's more water. So water is going to want to move out of the cell, which makes the cell shrivel up. So if you look, this is an actual microscope picture up here. You can see I have a bunch of shriveled up cells from the solute leaving it. And then finally, the last example, remember a hypotonic solution. That's where there's less solute in the solution. So maybe I have a cell that's super, super concentrated, and then there's almost no solute out. Here, that means there's more water on the outside, so water is going to want to move in, and it makes the cells swell up and actually eventually pop. And when it pops, this is called cytolysis. See that vocab word down there. And you can see in this microscope picture, my red blood cells look ridiculously huge and puffy compared to the original. So obviously, osmosis is, could really have a bad effect on cells. We don't want our cells blowing up or shriveling. So we have to have a way to make sure too much water is not moving into or out of our cells. So animals deal with this by making sure their cells are surrounded by a fluid that's isotonic. So this is part of homeostasis. Your body works really, really, really hard to make sure that the fluid surrounding your cells has equal salt concentration as the fluid inside your cells. Otherwise, your cells could burst. So like the plasma, the liquid in your blood that surrounds your blood cells, is isotonic to your blood. And also the extracellular fluid that surrounds all the other cells in your body is also isotonic. Plant cells deal with this by having a really strong cell wall that can't burst. So even if too much water goes into a plant cell, it will not burst because the cell wall is strong and rigid. And then some unicellular organisms, so things like paramecium, which are little unicellular protists, have what's called a contractile vacuole that basically pumps out any extra water that ends up in the cell. So let's look at this plant example a little bit, because it's kind of interesting. So if I take a plant cell, and I put it in a beaker of distilled water. So this is perfectly pure water. So you can kind of imagine I have pure water in this beaker. Because there's more water on the outside and there's some salt on the inside, water will move into the plant cell, causing it to swell. But because it has this nice and strong cell wall, it won't burst. And we call this pressure, the pressure of the water pushing on the cell wall, we call that turgor pressure. And if I put a plant cell in a solution with lots of salt, that means I'm going to have more salt on the outside, so the water is going to move out, and it makes the cell membrane kind of shrivel in. But because the cell wall is nice and strong, it still remains its plant cell shape. But it might make the plant wilt a little bit. And this is called plasmolysis, when the cell membrane kind of shrinks away from the cell wall because of the lack of water in the central vacuole. So, you can really see this effect when you water plants. So if you water a plant, you're basically adding water to the outside environment. You're creating a hypotonic environment, which means that there's more water outside the plant, so the water moves in, swelling the plant cells, creating turgor pressure, and making the plant stand up nice and straight because all its cells are really inflated. And kind of in the opposite, if you have not watered a plant, there ends up being much more solute and very little water on the outside of the plant. So water leaves the cells, making the cells shrink, resulting in plasmolysis when the membrane shrinks from the cell wall, losing turgor pressure and causing the plant to wilt. So we can look at this under a microscope. So you can see here's one plant cell that has been watered nicely, and you can see, the cell wall is really puffed out. The plant, we're getting a lot of turgor pressure, allowing the plant to stand up straight. And if you haven't watered a plant, water leaves the cell membrane shrinks in, and we get a plant to wilt. So to summarize, if a cell is in an isotonic solution, I have equal amount of water moving in and out of the cell. If a cell is in a hypotonic solution, that means there's more water on the outside, so water moves in, causing cells to burst, or if they're a plant, to have turgor pressure. And if we're in a hypertonic solution, there's less water on the outside, so water moves out, causing a shriveled cell or a plasmolyzed cell if we're in a plant. So that's it for video 6, and I'll see you in video 7.